I'm really excited to introduce Ethan Weiss, who I think I met at a Gordon conference on a hike in like 2008 or something. It's fantastic to have you here. Too bad it's not in person. So about Ethan, he went to Johns Hopkins University for medical school and stayed for internal medicine residency there. He then went on to UCSF for cardiology fellowship in a novel research track position where he joined the laboratory of Sean Coughlin studying thrombosis in the PAR receptors, the protease activated receptors. And it's important to note this is also the same environment where our current division chief got his scientific training. Dr. Weiss then went on to complete clinical fellowship in cardiology and then started as faculty at UCSF with his own laboratory that studied sex hormone mediated differences in thrombosis. And during this time, they developed a mouse model that had very interesting liver and metabolic phenotypes. And actually this inspired Dr. Weiss and everyone in the lab to change the research direction into metabolism and liver physiology. And this is actually pretty fantastic about being a fearless scientist and letting the data dictate the direction of the lab. Anyone who's been involved with Twitter knows that Dr. Weiss is very active on this platform. He's an ambassador of rational thinking and is also an excellent storyteller. He definitely embraces the fact that telling stories is how we communicate in both science and medicine. And so I'm sure he's going to deliver an excellent talk titled, "Is there an easy way to lose weight? Does it matter when we eat? Does it matter what we eat?" So without further ado, Dr. Weiss, please take it away.
Well, Alec, thank you so much for that amazing introduction. And I do remember that hike. In fact, I think I've got a picture of us at the top of some mountain in New Hampshire or something, different days. I appreciate the nod to storytelling. I love telling stories. I'm terrible at titles, so I'm going to skip on to the next one. So my disclosures are relevant disclosures, I guess, are that I'm a former advisor and I guess current stock owner of a company called VertaHealth based here in San Francisco, and I'm co-founder and advisor to a company also based here called Keto Inc. I'm not really going to talk about any of that work today, but I thought I'd at least mention it. So I'll start with a case. This is a real case, a woman, 63-year-old woman with hypertension, hyperlipidemia, and coronary disease. And she has a BMI of 38 at the time. And she'd been trying, like a lot of my patients, to lose weight unsuccessfully. And at that time, my pat answer to her and to everyone else was, when they'd ask, what do I do to lose weight? I'd say, well, just eat less and exercise more. And that worked about as well as telling my 4-month-old puppy not to eat the steak sitting on the ground. People were very frustrated by how poorly that worked. And so of course, she continued to gain weight. And then she came in one day and said that she had heard about this new thing called intermittent fasting and she wanted to know more. So I'm going to tell you a little bit about how we got into this. And Alec, you know, I appreciate the sort of nod to one of the things I love about biology and about our ability to spend time doing science, which sometimes feels like you shouldn't even get paid because it can be so much fun, is that you kind of go where the science takes you. And so while we did make this shift from thrombosis to metabolism 10 years ago, we've sort of made another shift now from doing mostly mouse-based physiology work to doing some human-based interventional nutrition. So I'll tell you about that today. So first objective is just to understand the importance of randomization. I don't have to tell this group. We'll talk a little bit about how to design a proper control group in nutrition science. I don't think there's a consensus about how to do that. It's obviously challenging because you can't be blinded. At least I can't think of a good way to blind people to what they put in their mouths. The second objective is to understand the efficacy and maybe even the safety of what's called time-restricted eating on weight loss and metabolic health, and I'll define what time-restricted eating is. And then I was told I needed to do this key points. It's kind of the same. So nutrition's a powerful tool. It can impact cardiovascular health, infrequent randomized trials, proper controls. And one of the lessons that we've learned is what happens in mice may not always happen in people. And so for next steps, aspire to do well-controlled and rigorous trials, temper excitement of preclinical or non-human studies, or at least use the non-human or preclinical work to try and inform biology as opposed to trying to explicitly link what's going on in a disease state. And then regarding intermittent fasting or time-restricted eating, potentially in the future, we'd like to explore interactions between meal timing and diet composition, and aspire to evaluate the effect of lifestyle on, and I wrote this as hard-ish cardiovascular outcomes, because while I think we would love to do hard outcomes, we can do better than what we've been doing. So maybe we can talk a little bit about that at the end. So I love to start off with this slide, which is a electron micrograph of a human adipose tissue. And I like to think about what the purpose of adipose tissue is. I think we all appreciate that it serves at least two basic functions. One is to insulate us from weather, typically Boston-like weather as opposed to San Francisco weather. And then the second is it really is the primary energy storage vessel. And I think one of the things that we've focused on in the lab, not yet as much in people, but is trying to understand some of the signals that govern the switch between energy storage and energy utilization. So that is, how does the body know when to turn off the storage or synthesis part and to start spending energy? And I like to use this slide as a way to point out two things. One is all living organisms share two common features, the first of which is the ability to reproduce, and the other is the ability to acquire, store, and utilize energy. And the other reason I like this slide is because I think it illustrates one of the things that'll be relevant to our conversation today about fasting, and that is that we didn't evolve as mammals with refrigerators and processed food or vending machines. That is, that we ate when food was available. And so we had to have mechanisms to be able to survive long periods without it. And so it raises the question that gets asked a lot when it comes to things like intermittent fasting, which is do we live in sort of a maladaptive environment today? And is the sort of natural or ancestral way we're supposed to eat more intermittent? And again, I'm just asking that as a sort of straw man. So what are the regulators of weight? I tell my patients now that we are somewhat like glorified automobiles. And again, I think there may be some nuances to that, but in essence, the most important is how much energy do we take in, and balanced by how much energy we spend. We spend a lot of energy just at rest, so doing relatively nothing. We're doing a lot of that these days. And then there's a sort of active energy expenditure that one might use, when you're out exercising. There's questions around macronutrients. Again, I won't touch on this today, but there are questions about whether altering the amount of, say, protein or fat or carbohydrate might have an impact on weight gain, particularly through its impact on hunger. And then the last one is this question about timing of food consumption, and I leave that as a question mark here. So there has been a fairly decent amount of work done on this in the observational epidemiology world, mostly in populations of people who practice fasting for religious purposes, especially Ramadan. It's been studied, I think, quite extensively, but there are other religious practices for fasting. And I think it's been questioned about there's been a long-standing question about the relative health benefits of fasting. And there's also been a important, I think, conversation around the relative health benefits of breakfast eating. Sometimes when I give this talk, I talk about sort of what your grandmother told you things. So, always eat a big breakfast and always clean your plate and things like that. So that's sort of one of the questions. So the overarching question that we sought to answer, and I'll tell you how we got here, was can by altering the timing of food consumption, can that be used as a tool to improve metabolic health? And I'm just showing you, these are old. I'm sure if you went on Amazon today, you could find another 300 books like this, but there have been a lot of people out there writing about intermittent fasting as a tool, metabolic tool or health tool, for the better part of the past decade. And I think what I'll tell you here is that that's been based on data that comes largely from animal models. So there was work done by Sachin Panda at Scripps in Southern California, starting off in flies, talking about time-restricted feeding as a way to improve health. And I'll tell you more about Sachin's work in a minute, but all of these books were written, at least to my knowledge, before the publication of any trials in human beings. So let me define what we're talking about. So time-restricted eating is what we call when we do it in people, and time-restricted feeding, since the animals don't really get a choice, is the term used in animal studies. And they fall under the umbrella of intermittent fasting. So intermittent fasting just means altering the normal amount of time between meals that you would have without thinking about it. I think it's sort of a funky definition, but if you think about it, and I think there have been studies that have looked at this, that people tend to eat almost every hour that they're awake. I think the average number of hours that a human being in the United States eats is 14 a day, which is a lot. So if you shorten that eating window, you increase the fasting window, and that sort of defines intermittent fasting. And there are hundreds of different forms of this. I've got a patient of mine who does twice a year a 1-week fast where he has nothing but water, and then on Sunday he breaks his fast with a steak and a glass of red wine, and then he does the same thing again. He probably is the only one who practices that form of intermittent fasting, but there are many, many different forms, ranging from sort of shorter fasts, just even like 10 or 12-hour fasts, up to multi-week fasts. So again, altering the time of food consumption. The most common of these is so-called 16-8. So that means you fast for 16 hours and eat for 8 hours. And I'll tell you two different forms of that, and we'll describe one that we tested. So this story really got traction back around almost 10 years ago now, when Sachin Panda and his group published this first paper looking at the effect of time-restricted feeding on metabolism in mice. And so I just want to spend a minute going through the design here, because I think it's really important, and it informed a lot of the work that we ended up doing, and actually some of my own practice. So the idea here is that the mice were randomized to either receive a normal diet, which is normal mouse chow, or a diet that was high in fat. And again, this is so-called high-fat diet in mice. It's actually high fat, but also extremely high in sugar. And they were then randomized within that to either be able to eat whenever they wanted to. And again, mice are nocturnal, so they're eating mostly during the night, but they do eat and graze during the day. Or they were given the exact same amount of food, but it was only given to them during a 9-hour window. And so again, so 4 groups: normal chow, ad lib, normal chow, time restricted, and then high fat, ad lib, and high fat, time restricted. And importantly, what they saw was that the mice in all 4 groups ate the exact same number of calories. So that helps in interpretation of the results, that there was no difference in calorie intake between any of the 4 groups. But what they found, and I think quite strikingly, was that while the groups that were randomized to this sort of normal diet didn't seem to have too much of a difference between this time restricted and the ad lib, when you look at the animals that got access to this high fat diet, or high fat and high sugar diet, there was a tremendous divergence in the curves happening very early between the animals that were given their food only during the 9-hour window here in red, versus these animals in whatever this color, peach or whatever it is, that were able to eat all the time. And you can see a representative image of what the 2 mice look like. And it wasn't just that they lost a lot of weight, they had improvement in other metabolic parameters, including fatty liver. So you can see here on this tiny little H&E slide that the animals in this FA group, so these are the animals who were able to eat ad lib on the high fat diet, had rip-roaring fatty liver, whereas the ones in eating the same amount of food, the same calories, but only during this 9-hour window, had protection. And what I want to point out here is that in doing this work, the Panda group latched on to an observation that I think was really important, which was sort of trying to understand what is the mechanism of benefit here. So these animals are eating the same amount. Was it that they ended up exercising more? Again, if you go back to the sort of car model, was that were they spending more energy during exercise? And it turns out they didn't. Their activity levels were the same, but they did have an increase in their resting energy expenditure. And so this kind of took hold immediately as kind of the perfect weight loss tool, right? You basically eat the same amount of food, you don't have to exercise anymore. You just basically turn up your idle, your rev, you burn more at rest, and all you have to do is basically narrow your feeding window. So it became really exciting to the field, and I will say personally exciting to me. They then went on to ask if you could not prevent metabolic disease, but if you could fix metabolic disease. So they took existing mice, obese mice, and then did the same experiment. So I'll show you this. This is a little more complicated because there are 4 groups, but if you just pay attention to the top 4 here, I'll define them for you really quickly. What they did was a crossover study here. So this is high-fat diet. This blue line here is high-fat diet, ad lib, and they stay on ad lib, and you can see they continue to gain weight throughout the course of the 36 weeks. The red line is this high-fat diet group, ad lib up until this 25-week time point, at which point they're switched over to time-restricted feeding. And you can see that they then start to fall off on their weight gain, and it improves dramatically. And they did the exact same thing in the opposite directions. They took mice that were getting high-fat diet on time-restricted, and they had a very modest weight gain. And when they switched them over to being able to eat ad lib, they started to gain weight. And so same thing here in terms of liver fat content. So again, fatty liver, metabolic disease. And so again, this got a lot of attention in the press. And I'll just show you a couple of examples. This is from 2015, 12-hour window for healthy weight. This is 2016, my friend Anhat O'Connor, fasting diets are gaining acceptance. More recently, intermittent fasting made my life easier and happier. And then Jane Brody just last year, about a year ago, the benefits of intermittent fasting. And again, I want to emphasize that up until this point, there had been a lot of human clinical trials that were ongoing, but none that had been published, or at least none had been published with a control group or an adequate sample size. This is an example of a study that was published recently before our study came out. This is also from the Panda group, and it was in 2020, so last year. And I'm not going to go through details of this, but the punchline was 10-hour time-restricted eating reduces weight, blood pressure, atherogenic lipids in patients with metabolic syndrome. And I just wanted to point out here that this important point, which is in a single arm paired sample trial, 19 participants. So again, relatively small sample size and no control group. So all of this, I'll just pause for a second and say what I said before, which was that I got caught up in this excitement myself back 2013 or so, and started trying to do this 16-8 time restricted eating. And I think I lost some weight. I think I lost 5 or 6 pounds and found it to be not that difficult. And again, based on all of the sort of questions of maybe we're living in a different environment now, and maybe this is maladaptive, and gosh, what a great intervention if you can, lose weight without having to do anything except basically skipping breakfast, and it revs your energy. And there were all kinds of stories now coming out about how people felt better, and it just seemed like it was all too good to be true. But that said, we decided, you know what, we ought to, at least before recommending this to people widely, we ought to at least design a study that we can feel good about. So we set out, probably 4 or 5 years ago to design this little study. It was initially a pilot. It turned into more than a pilot. I'll walk you through it now. So I should mention at this point that this work was done entirely, I shouldn't say entirely, 95% of the work was done by a former graduate student in my group by the name of Dylan Lowe. Dylan was in the BMS graduate program here at UCSF and had come to my lab to do a rotation, and didn't choose my lab, went to a different lab, but came to me one day telling me that he was going to leave graduate school because he wanted to do more human and translational work. And so I said, well, instead of leaving graduate school, why don't you come and we'll do a clinical trial together. I've never done one before. And he pulled it off and did a phenomenal job. So I just want to highlight that. So this was our aim, was to determine the effect of time-restricted eating on weight loss in overweight humans. We designed a prospective randomized trial, and so we randomized people to time-restricted eating versus control. I want to spend a moment here talking about control, because I think we all understand the importance of having a control in our experiments, and this is no different. Of course, in this world, designing a control is not easy, and so we spent some time thinking about what is the proper control. A lot of times in nutrition studies, you'll see people get randomized to the control group, which is basically they'll get handed a pamphlet and get a pat on the back. And we felt like, and again, I think this is probably open for debate, but we felt like, since it's an unblinded by definition study, that we wanted both groups to feel like they were getting an active intervention. We didn't want people to feel like they were getting placebo. We didn't, and we didn't want that because we didn't want people to drop out, but we really wanted to test the effect of the intervention, not people's perception of whether they got assigned to an active intervention or a control. So with that, we basically came up with an, I don't know if it was deceptive, but at least a somewhat tricky way to convince people that the control group was active. And so we called it consistent meal timing, or aligned meal timing. And what we did was we gave people a window, and I'll explain in a second, of when they could eat. And it basically was all day. And so the primary outcome for our study was a change in weight. But we looked at a lot of other outcomes, secondary outcomes, pre-specified ones, including fat mass, lean mass, insulin, glucose, hemoglobin A1C, et cetera, et cetera. But we also did a tremendous number of exploratory outcomes, which I can talk about. We recruited beginning in August of 2018 and finished it in just over a year. And I'll tell you a little bit more about the design. So there were two things that we ended up actually starting off with: a smaller study where we were going to bring people in and do very intensive metabolic phenotyping on them, where we would do pre-post measurements, measuring energy expenditure, both using doubly labeled water to measure total energy expenditure, but also doing indirect calorimetry for resting metabolic rate, multiple different kinds of body composition analysis, bloods, metabolomics, everything you can think of. And then we also decided that we would add another group of patients in whom we would only send them a scale and let them download this custom app that we had designed. And we were able to then sort of increase our sample size. So our inclusion criteria had to be an adult. We included males and females. BMI was between 27 and 43. And at least for the in-person, you had to be within 60 miles of UCSF. For the purpose of the study, we asked people that they had to regularly consume breakfast. So we didn't want people who were already practicing some form of intermittent fasting. And then we excluded all the sort of obvious things. We didn't want people with diabetes, and we didn't want people who were unable to fast. And then at that time, it seems kind of quaint now to think about this, but we excluded people who frequently traveled across time zones, or people who had unusual work hours. And so here was the basic overall design. Again, 12 weeks, which is, I think, the sort of bare minimum for a nutrition study. A consistent meal timing group. They were able to eat their first meal between 7 and 11, their second meal between 11 and 3. They had a 1-hour break, and then their third meal between 4 and 10. We told them they could snack as much as they want, and so we didn't tell them anything about what else they should or shouldn't put in their mouths. And then the time-restricted eating group was only water or black coffee or tea, so non-caloric beverages without sweetener, between the hours of 8 p.m. and 12 p.m. And then between 12 and 8, they were told they could eat whatever they want. And again, they were told they can eat whatever they want. We didn't tell them that they had to change their diet in any form, that they couldn't snack. So between that, in that window of 8 hours, between 12 and 8, they could eat whatever they want. And so again, this was designed in collaboration with this group, Healthy Heart, or Eureka, here at UCSF. And they designed us a custom app, which was able to deliver some surveys. They did their daily weights through a wireless scale, blood pressure, reminders, adherence questions, et cetera, et cetera. And so this is the in-person measurement timeline. So again, people came in at -1 week to our CTSI, and we did this total energy expenditure analysis. So this was the first of the double A labeled water that they drank. And then they came back at time 0, had some blood draw, body composition, resting energy, the second part of the TEE analysis. And then they got started on their diet, 12 weeks, and they came back at, again, -1 week, so week 11 to do this first TE repeat. And then the final visit included all the same measures. So this is what things looked like. And again, for the virtual people, they just got their scale and the app, and they were off to the races. So this is the paper. This came out in the end of September. Paper got a lot of attention. It's surprising for what I'm about to tell you was a negative result, but it did generate a lot of enthusiastic discussion. So this is our consort diagram. So we had almost 2,000 people who were assessed, ended up randomizing 140, 70 to continuous meal timing, and 70 to time restricted eating. We lost a sort of expected number of people over the course of 12 weeks, ended up with 57 and 59 in the two groups respectively. So this is what the baseline looks like, and I won't go through everything here, but I just want to point out this is a relatively young group of people, young at least by my standards these days, so 46, roughly 60% males, which is unusual for weight loss studies. Usually it's a predominance of females. And to make it really easy to understand the math for all of us, the baseline weight was roughly 100 kilograms in both groups, with the BMI roughly 32.5 or 33. Within the in-person cohort, you'll notice the only major difference was that it was a little bit of a younger group. So again, these are people in the Bay Area as opposed to the entire United States. The other thing that is important is that it looks like their weight was they were a little bit lighter, so 93 kilograms and a BMI of 31 versus 33. So this is self-reported adherence by the app. And what you can see is that people were able to do the continuous meal timing as one might expect, because it really wasn't a challenge, and that people were largely able to at least report that they were able to stick with the time restricted eating throughout the beginning of the study, and there was some drop off towards the end. So these are the primary data. This is each one of these dots is an individual person stepping on their scale in their own home. And then this is a linear mixed model to look at the treatment effect. And what you can see here is that both groups did lose weight. So the blue line here is this continuous meal timing. The orange line is the time restricted eating group. But there didn't seem to be a gigantic effect either within the groups or between the groups. And I'll show you the data here. So again, within the total cohort, the mean starting weight was 99 kilograms. And in the continuous meal timing, they lost a little over a half a kilogram. It was not quite statistically significant within the group. Within the TRE group, again, started off at 99. They lost almost a kilogram. That was statistically significant, but within groups, there was no statistical difference between the amount, between their weight. And then this is the percent weight change. And again, a significant decrease within group for both, but no between groups difference. And so this really is the primary endpoint, which was negative. This is a waterfall plot, which is something anyone who's looked at weight loss studies will be used to seeing, which is each individual in the study, and it's the percent of weight that they lost over the course of the study, the entire 12 weeks. And so you can see there was one person here who lost what it looks like 6.5, 7%, a large number of people who lost a little less than 5%, but really only one person who lost more than 5% of their body weight, which is kind of the standard of, is this an intervention that actually is worth anything? And then, you know, a little less than a third or so gained weight. And this is the continuous meal timing group. And what you can see is that there seems to be an ever so slight difference in the distribution of people. Is about 50-50 about people who gained weight and lost weight. But if anything, the people who lost weight lost more in our control group. So in particular, I was asked a bunch when I was interviewed during the time this paper came out. I was asked about, well, how do you respond to people who say this worked for them? Or how do you respond to people who say it worked for a celebrity? Like Jennifer Aniston is a huge, you know, proponent of time restricted eating or intermittent fasting. What do you say to her if she says it worked for her? And so my favorite retort to that was to say, well, what if Jennifer Aniston came to see me in the clinic and I told her I had this great new intervention called continuous meal timing, and she could eat between 7 and 11, 11 and 3, and 4 and 10. And she comes back in 12 weeks and she's lost 8% of her body weight. She's going to be really excited about this new thing, and she's going to ask me to write a book. So I think this to me was sort of a hint of how important it was to have a control group. And this control group, even though it wasn't designed to be active, it certainly was perceived to be active by the participants. So this is the same thing though, but just within the in-person cohort. I do want to go through some of these data because this is where we get to some of the meat of some of the things that we found that might have been a little bit unexpected beyond just the primary endpoint. So again, a relatively leaner group of people to begin with, so 92 kilograms. And within the CMT group, again, they lost about a half a kilogram. And there was a greater numerical amount of weight loss in the in-person group. So these people lost about a kilogram and a half. But again, there was not a statistical difference between the two groups, and nor was there in terms of weight change. I want to highlight the 1.6 kilogram number because I'm going to come back to that in a second. So we measured a lot of different things. Like I said, respiratory quotient, resting metabolic rate, total energy expenditure. I'll just highlight here quickly that total energy expenditure. So we were designed as creatures not to burn ourselves out. That is that when you start to lose weight, there's a compensatory decrease in resting energy expenditure that allows us to continue to survive. It's normal. It's an adaptive response to starvation. And as you can see in almost every human weight loss study, there's a decrease in energy expenditure with weight loss. And so these people did lose weight, and so therefore they did have a sort of a compensatory decrease in energy expenditure. In the CMT group, it was about 127 kilocalories per day. In the time-restricted eating group, it was about 185 kilograms per day. Kilocalories per day. And I wanted to just point out, while there wasn't any difference between the groups, if you remember back to when I was telling you about the first set of experiments done by the Panda group, one of the things they saw was an increase in energy expenditure relative in the time-restricted feeding group relative to control or to ad lib eating. And so I guess one of the questions that we had at the beginning was, well, maybe what you'll see is not necessarily an increase, but a relative decrease, or a relatively smaller decrease in energy expenditure in time-restricted eating. That is that maybe you won't slow down your metabolism quite as much. And I just want to point out that did not happen. So again, if anything, numerically, there was more of a decrease in energy expenditure in the time-restricted group than there was in the continuous group. And so this is important. I think we did extensive body composition analysis, and I'll highlight a couple of things here. So, So if you look at the change in fat, so in the continuous meal group, pre and post, there was no difference, basically no change. And in the time-restricted eating group, they did lose half a kilogram of fat. Now, if you recall, they lost 1.6 kilograms on average of total body mass. And so I'm going to preview for you what that predicts. And no difference in percent fat mass, visceral fat mass, or subcutaneous fat mass. So really just a very modest or mediocre, almost non-existent impact on fat mass. And this was something that we did not expect. And I think it ended up getting a lot of attention in the press. And I think it's worth at least discussing. And I'll say what I've said a lot, which is this is, to me, an interesting finding that was in an exploratory outcome that I think all it says is that we should look at this more carefully in the future. And so what we found was that so let me just stop for a second and say that in a typical weight loss study, if you lose a kilogram of body mass, on average, 1/3 of that, so 300 grams of that will be from lean mass, and 70% of it, or 2/3 of it, 66% will be from fat mass. So you're losing 2 for 1. You're basically losing two parts fat for every 1 part lean, which seems like a decent trade-off if that's what you're intending to do. We saw the opposite here. So here in the continuous or control group, they lost about 1/3 of a kilogram of lean mass, but in the time-restricted group, they lost 1.1 kilograms of lean mass. Now, again, I want to be careful. This is lean mass. really technically it's fat-free mass. We can't say that this is muscle mass, but we did see this signal with an appendicular lean mass. So appendicular lean mass is basically the amount of lean mass you have in all 4 of your limbs. And I'll talk about that more in a few minutes, but there was a statistically significant difference between the groups in the amount of appendicular lean mass loss. So it was 0.64 kilograms versus 0.17. And this was something that was, I think, notable and important. I won't go through these, but there was no change in any of these other relevant metabolic parameters, that including glucose, insulin, hemoglobin A1C, any of this stuff, LDL cholesterol, any of it. So really no impact there. So the take home for me was there was a trend, or trivial trend, towards greater weight loss in the time restricted group versus control at 12 weeks. I think the magnitude of weight loss in either group was trivial. And so I think, we can get caught up in some experimental design issues or this or that, but the fact is, whatever you want to say about the control group or what they might or might not have eaten, no one lost a lot of weight in this study. In fact, if you look at the total amount of weight lost, it ends up being about 0.2 pounds per week, which is really not very much. And I think, what I'm comfortable saying is that most of the weight that was lost, at least in the in-person group, came from not fat mass. And I think that's important. So in other words, people will sometimes ask, like, well, what about it? What if your aim is to not lose weight? You just want to improve your body composition or your metabolic health. Well, at least the way we designed this study, we couldn't really say that there was any positive change in metabolic health, and the body composition changes seemed unfavorable. So we'll do some other exploratory stuff in the future, and we have some follow-on experiments. But the bottom line is that TRE is really not effective. And I say this very carefully and hesitantly, but I think there's a potential for harm, especially in older people. And I want to discuss that in a second. And again, I understand that this is super preliminary and really just hypothesis generating. I will say that when we unblinded these results and analyzed the data, I stopped doing time restricted eating myself and haven't now for over a year. So let me tell you a little bit about appendicular lean mass. So turns out it's very well studied and it predicts mortality. I'm not going to go through a lot of data, but it looks like that for each increase in appendicular lean mass, there's a 50% lower risk of mortality. So I think it makes sense that if you have more, and again, who knows what the chicken is and what the egg is. I'm not going to try and imply causality here, but if you're an older person, you've got more muscle mass, it probably means you're healthier. I just wanted to quickly walk through a couple of potential explanations for why there might be a difference in lean mass. And these are all just sort of off the top of our head guesses. So one idea would be that maybe there's a difference in intake of protein. There are data suggesting that people tend to eat the same relative amount of protein with each meal. And since they're eating on average one fewer meal per day, maybe that they're just a little bit protein restricted. There's also some interesting data. I'll show you a little bit about that now, about protein timing, and I'll come back to that one. There's this question about meal timing. So I mentioned earlier that there are really two forms and probably more of time restricted eating. In the last 4 or 5 years, there's a fantastic clinical investigator at UAB named Courtney Peterson, who studies what's called early time restricted eating. And I'll show you some data on that in a second, or maybe there's something else altogether. So what about this idea of protein timing? So if you think about it, right, we are circadian animals. We do have hormones that get secreted at different times of the day. And a lot of the hormones that seem to be important or relevant to protein synthesis do get secreted at night or early in the morning. And so these guys did this experiment that I thought was really nice. They took people and they brought them in. They did muscle biopsies on them at the beginning, at the end. And they designed a really neat study where everybody was isocaloric. So you can see here that everyone got about 2,400 calories. These people were fed their meals. And they were either given it all throughout the day, so even, so 840 in breakfast, 820 at lunch, or 700 at dinner, or they skewed it to later in the day. So here they were relatively having fewer calories in the morning, 500 versus, and then 600, then most of it at night. And you can see the same thing happens here for protein. So it's 10 grams or so in the morning, 16 and 63. So most of their protein at night out of a total of 90 grams. And what they found was, you can ignore the day 1, day 7 issue, but in the what they found was that there was a decrease in the synthesis of new muscle in people who were in the skew group, so the people who had most of their protein at night. And again, I think this is all super early and preliminary hypothesis generating, but there is something I think potentially there about having your substrate around for when your machinery is revved up to make new muscle. And again, like many processes in biology, we're constantly making and breaking down muscle. It seems like a fetal cycle, right, where you'll make it and break it down, but we do that. It's probably part of a normal homeostatic regulatory mechanism, but it turns out that a lot of the synthesis may happen earlier in the day. This is a little bit on this distinction between early and late time-restricted eating. So early time-restricted eating, again, our study was you could eat anything you want between 12 p.m. and 8 p.m. This is a very similar sort of cousin to that. The difference being that they could eat whatever they want between 6 a.m. and 2 p.m. And it sort of is, I think, somewhat based off of this idea that maybe you want to have most of your calories, whether they're protein calories or NE calories, earlier in the day. And so Courtney Peterson and her group have been working on this for years. This is a paper that that just came out, and I'm not going to go through the details, but what you can see is that they did see some improvements in metabolic parameters, particularly in insulin, and they actually just presented in November at the Obesity Week meeting a randomized trial, sort of smallish but decent size. I think it was 70 or so people, looking at the effect of early time restricted eating versus a control. Now, what was interesting here was that the people, so it was 90 people, the people randomized to control were randomized to to the normal UAB outpatient weight loss clinic. And so what you can see here is, it was 7 a.m. to 3 p.m. And 14 weeks. And what you can see here is that there wasn't any change in weight. So, but importantly, both groups lost a lot of weight. So I think, let's see, it was 12 versus 10% of body weight. And that is because the control group in this group was very active. This is a really nice, well-designed study. Didn't really show any differences except maybe a change in abdominal fat and potentially some effects on blood pressure and insulin. So a lot more to learn here, but I think this is an intriguing possibility for the future. So for our future, we've recently submitted a new grant application to evaluate if there's an interaction between meal timing and diet. So we're going to do a 2x2 factorial of time-restricted eating versus continuous meal timing. And again, we'll then randomize to low fat versus low carbohydrate, and that'll be kind of fun. The idea being that low carbohydrate diets may have benefit if they do through effects on hunger. And then again, I think there are some questions about meal timing, which we'd like to explore, and protein timing as well. So I'll just stop and mention all of the people. Again, I just want to highlight Dylan in particular. Division of Cardiology was very supportive and helped us get this thing off the ground. And I guess with that, I will stop and save some time for questions.
Wonderful. Thank you so much, Dr. Weiss. I think we have a couple of questions coming in on the chat. In the meantime, everyone can please feel free to come off of mute to ask something directly. We have a question from Dr. Warren Manning, who says, excellent presentation. Is it possible that not one approach fits all? There are some individuals for whom continuous eating leads to weight loss, and for others, a time-restricted eating format is best. The problem is how to identify which group you fit into.
It's an outstanding question. I think there's a greater question of sort of what's, if there's such a thing as precision nutrition. I know the NIH has a huge interest in this. I guess if you go back to like the waterfall plots, there's no doubt that it worked for some people and it didn't work for other people, but so did the control. And so I think that, I guess I'm curious. I mean, obviously we're all super curious about why some things work for some people and they don't for others. I'm not sure I'm equipped yet to answer that question. I'm not really sure how I would design such a study, but it's a really good question.
We have a couple more from Dr. Callan Ho. So is it better to eat breakfast or skip breakfast?
Well, I'm not going to make this mistake ever again. I got in the habit of telling people that it was better to skip breakfast. Because I thought it was like a contrarian thing that like our moms all told us to eat breakfast. But I'm not biting again. I'm going to tell you, I don't think there are good data to support that there's an answer to that
Hold
question
on.
that I would believe.
Hold on.
Sounds like somebody's having breakfast.
Yeah. I'm trying to mute. Just one moment.
You stop.
If someone could please mute their phone, having trouble locating it. Okay, I think we're all set. So I know Rob has a bunch of questions. I have one more question on the chat from Dr. Dan Katz. Thoughts on TRE and longevity given a reduction in resting metabolic rate?
I mean, I guess the question is there an effect of TRE independent of calorie restriction? And I think, you know, we have to probably limit our conversations about longevity to non-humans, or at least probably non-humans. I don't know of any data yet in mice to show that there's an increase in longevity that's calorie independent. I'd have to go back and look for sure. Sachin may have published something in flies, but my guess is that there's probably not going to be a huge impact. I know everybody wants to believe there's like, you know, favorable impacts on TOR signaling and all this other stuff, but I'm not sure I'm there yet. And again, at least the way we designed this real world study, there wasn't a huge difference in calorie intake, resting energy expenditure, or weight. There really not that much change.
Rob, you had a couple of questions?
Yeah, that was fantastic. Really a thoughtful talk. So at the beginning of the talk, you kind of reminded us that our evolutionary job is just to procreate, right? To get food and to use it to procreate. And women and men have become, for a million hormonal reasons, are very different in that regard. Have you looked at men versus women? I can tell you the reason we're interested is we've looked at a lot of this chronic exercise data, and we see striking differences in effects across various parameters between men and women. I realize, look, it was heroic just to enroll as many patients as you did, but any hints between men and women? That's my first question.
Yeah, I mean, look, Rob, I think it's a great question. Our study was so small as it was. I'd hate to like start slicing and dicing the data even more. We've not done an analysis of sex. I will tell you this though, having spent a lot of time in this space, there are strong anecdotal data that women, especially postmenopausal women, tend to have a harder time with weight loss. And it's enough of a signal in the anecdotal noise that I think that it's worth looking at, but we haven't done it yet, but would love to.
So the other question I had is, I know we've discussed this before related to the study that you already did, but moving forward, how are you going to control for the amount and the quality of the food that people are eating, right? Because you're not giving them the exact same meal, correct? So unlike the C57 black 6 mice that you can put in a cage and give them all the same pellets, here you're just going to be restricting the time, or you're going to be controlling what they get too, you know? So
that's a great point. And I should have made this clear. So in the study that we did, which was really a real world prescription, right? And we did not have any idea what people were eating. We didn't even ask them what they were eating. We didn't really care. And the purpose was to probe whether there was an impact of just telling people to do this, with the idea being that it's a pretty simple intervention. And if it works, hey, that could be something that could be applied to a large number of people. I thought it was really interesting, but as you say, the scientist in me wants to be a little bit more able to understand what we're actually doing. So for this proposal that we just put in to the NIH, we're going to do a hybrid sort of in between the gold standard of admitting people to a metabolic ward, where you actually have a metabolic kitchen. You can feed them exactly the same, you know, what they eat, you measure it, you can measure their, you know, stool and their, you know, urine and everything. So we're going to do a hybrid between that and the real world prescription study we did, which is we're going to deliver their meals. There's a company that will actually function as sort of a quasi metabolic kitchen and sends out meals once a week.
Gotcha.
We'll instruct them to eat nothing but the food that we give them. and we'll have them weigh what's left over. So it'll be kind of a hybrid approach. And hopefully we'll get a little bit of a better idea. And the reason we're doing that for that study is that we are hoping in that study to explore an interaction about diet itself. And so if you're going to do that, you really want to know what you're feeding people.
Great. We have a couple more questions that have come in through the chat. Dr. Mike Mee says, great study. In the TREAT trial, there was noted a decrease in physical activity level of the TRE group. Perhaps that could explain some of the difference in lean muscle loss or lean mass loss.
Very perceptive, Mike. Yeah, so again, in a small subset of the in-person cohort, we put fitness trackers on people. We had them wear Oura rings. It ended up being around 40-ish people, and there did seem to be a difference in physical activity. I'm gonna defer to my statistician friends and colleagues. This was one of our exploratory outcomes, one of hundreds. So I think it's interesting, and we probably ought to think about that, but that's about as much as I can say, I think, based on those data.
Great. We have a question from Dr. Julia Restia. Are there studies of sumo wrestlers who have good muscle mass and high fat? I think they eat most of their calories late in the day.
Wow, no, I'm sure there are studies of sumo wrestlers. I hadn't, I mean, I think that there's a lot of work being done in this sort of so-called metabolically healthy obese population. So people who have a high BMI and a high fat mass, but have normal metabolic parameters. I am sure there's got to be work done about sumo wrestlers, but I'm not familiar with that literature at all. But it sounds interesting. I'll have to look.
Great. Dr. Jeremy Robbins was also asking about the physical activity, but also says, understanding your study was small, did you look at the extreme responders and non-responders in the CMT group to see what may have contributed to those vast differences?
We haven't yet. It's a good idea. You know, I worry, just given the size, that we may not get too much out of it, but it is interesting. I think it's generally interesting, you know, that somebody can lose as much weight. There is this thing called the Hawthorne Effect, which is, I think, I hope I don't get this wrong, but I think it's basically if you hand somebody a scale and ask them to step on it every day, they'll lose weight. And so I think for nutrition studies, basically perceiving that you're in a weight loss study is enough to make you lose some weight for some people.
Great. A question from Christine. Sorry, all I have is iPhone, not the last name. Any plans to perform a study with a smaller daily window, for example, a 2-hour window versus an 8-hour window?
I mean, I would love to. This is the problem, right? You could design almost any permutation and combination of different things. One of the things that sort of drew me into this world was trying to find solutions for my patients. And one of the reasons I think that people have such a hard time with weight loss is that it's very easy to fail, and that it's often too hard. And so I've been thinking, so what are things that we can do that are going to be relatively easy for most people, or a lot of people, but also where people are going to feel like they're successful. And so I feel like shortening a feeding window down to 2 hours. I mean, I certainly could do it. I've fasted up to, I think, 48 hours before. Yeah, there are lots of things we could look at. But I think right now I'm trying to keep it in sort of what are the things that people can probably do practically.
I had a quick question about some of the big weight loss programs like Weight Watchers or Noom, and given that folks are now pretty easily entering what they're eating on an app, granted not always with full fidelity, is anyone looking at metabolic changes associated with those big programs, which presumably have a lot of participants?
So Weight Watchers has an enormous amount of data around its program over the years. Most of it was generated back when Weight Watchers was mostly in person. So, when people would come in, I think there's been less in the virtual world. I don't know how much Noom is published. It's a really good question. We are, so in my other world as a sponsor for this little company that we started, we are gonna be publishing a study, a head-to-head comparison of this low carbohydrate diet that we designed versus Weight Watchers. and I can't talk about it yet, but that should be something that will show up in the next few months and will be interesting. How about that?
Looking forward to that. A couple more questions from Warren Manning. Historically, when did man start eating 3 meals per day? And what are the time eating habits of indigenous tribes in Africa untouched by the Western world?
Great question.
Phenomenal questions. And the first thing I'm gonna do when I get done here today after I talk to Rob is to go and see if I can find out the answers. I don't know. I'll have to ask or look and see if I can find. Those are great questions. I just don't know the answer.
Wonderful. This has been a really great discussion. Thank you so much, Dr. Weiss, for joining us. And if there are no further questions, we'll see everyone at Grand Rounds next week.